Semester 5 Paper 10 CBCS syllabus is Microbiology, Plant Pathology and Mushroom Culture Technology. In this unit number 1 is Microbiology. Today we will see about sterilization methods. Definition: it is a process in which all the living organisms are killed. Sterilization means anything living or non-living or any medium made free from all microorganisms, including virus, bacteria, fungi and their reproductive structures. Spores, which are pathogenic or non-pathogenic. The microorganisms are present in the nature, contaminated every medium. Pathogenic microorganisms become harmful and cause harmful diseases in human beings and animals. They are able to grow well under suitable environmental conditions. For avoiding the growth of pathogenic microorganisms, sterilization method is used in culture media. For the suspending fluid, reagent, containers and equipment used in microbiology laboratory, during various experiments, sterilization is essential. Medical and surgical instruments and all materials used to penetrate in the tissue of man and animals. For example, surgical operations, hypodermic injections and diagnostic target require sterilization. For pure culture, it is done by physical and chemical method. In this slide, we will learn about physical method of sterilization. Artificial or natural sterilizing agents are available. For example, sunlight, heat, moist heat, dry heat, flame, filtration, radiations. Sunlight play important role in sterilization. High intensity of light during summer used as sterilization agent. Glassware and equipment required in laboratory are sterilized under high intensity of sunlight and then used for various experiments. Then heat sterilization. Fire and boiling water have been used for sterilization and disinfections since the time of Greeks. Heating is one of the most popular ways to destroy microorganisms. For this heat sterilization, dry heat and moist heat is efficient and effective method. It kills microorganisms by coagulating and denaturing the nature of enzyme and proteins. Pathogenic spores are killed by exposure to moist heat at 121 degrees centigrade for 10 to 30 minutes. Then dry heat kills organism by oxidative destruction of cell and spores are killed by exposure to dry heat at 160 degrees centigrade for 1 hour. Moist heat sterilization or steam sterilization is prepared in the autoclave by heating the water. The instrument autoclave is used in laboratory for sterilization of glasswares, culture mediums. Heat sensor bacteria under various temperature range are killed by maintaining the temperature range. Fungal spores, viruses along with sterilizing material are killed by moist heat procedure produced in autoclave. Some bacteria, fungi, viruses are killed in 10 minutes at 50 degrees centigrade, while some viruses at 50 degrees centigrade for 10 minutes. Most of the bacteria require 121 degrees centigrade temperature for 10 to 30 minutes. This is autoclave used for the sterilization method, which includes pressure regulating device, pressure gauge, valve, autoclave lid, handles, autoclave body, steam release valve, vacuum release valve and stand. Then dry heat is used for sterilization. The atoms to be sterilized are placed in an oven at 160 degree centigrade to 170 degree centigrade for 2 to 3 hours, and the microbial death appears from the oxidation of cell constituents, denaturation of protein. Dry heat is less effective than moist heat, but it not corrode glasswares or instruments. It is significant for powder oil sterilization or petri dish pipette sterilization or plastic and rubber atom sterilization. Then flame is useful sterilizing agent in laboratory. Burning article in spirit of exposing it to spirit of gas flame. Scalpel, needle, forceps are sterilized with help of flame. This process doesn't produce high temperature for sterilization. The next method is filtration method. It is excellent way to reduce microbial population in solutions of heat sensitive material, toxin, serum, antibodies. Small filters are used for filtration, viruses, bacteria and mycoplasma. There are various types of filters used for the sterilization of solutions: asbestos, asbestos paper, disc, membrane filter, sintered glass filters, microfilters. The widely used instrument is laminar. Airflow is biological safety cabinet using in the different experiments as a sterilizing instrument. In this instrument, air can be sterilized by filtration, laminar airflow cabinets. It is mostly used in tissue culture laboratory. Filter size is different and with high efficiency particulate air filters which remove 99.9% of 0.3 mm particles. It is one of the most important material for air filtration system. Highly improved sophisticated inoculation chamber is used for antiseptic transfer for microbial culture or pure culture. It is designed in such a manner that sterile materials remain protected from contamination. In inoculation chamber, filtered air is flowed in single direction. Chamber is with glass filters, fiberglass filters, blowers, UV light, tube light fluorescent for illumination, glass doors. Let us see this figure. It is inoculation chamber with UV light and airflow system. Then radiations. It is energy that comes from a source and travel through space at the speed of light. It shows different types. Electromagnetic radiation consists of waves of energy with associated electric and magnetic field. Then ultraviolet radiations around 260 nm is lethal but does not penetrate dirt, film, water or other substance effectively. Because of this disadvantage, UV radiations is used as sterilizing agent in specific situation. UV light or UV lamps are used for sterilizing air. Many of vegetative forms of microorganisms are present in air and killed by UV rays. These radiations are used in sterilizing the surgical instrument in hospital, school, storage or warehouse. UV rays are produced by mercury vapor. UV rays sources are harmful to mankind. Then ionizing radiations are used for sterilization. These radiations include high-speed electrons, X-ray, gamma rays. X-ray, gamma rays are high-energy ionizing radiations with high penetrating power. Gamma radiations from cobalt-60 source is used in cold sterilization of antibiotic, hormone and plant products. X-rays are used to kill the bacteria in various mediums. Then the next method is chemical sterilization method. Chemicals are used in disinfection and it is antiseptic content. These chemicals are called as a disinfectants which have significant role in sterilization practice. The chemical must be toxic for infection agents. It should be soluble in water and lipids for penetration into microorganism. It should be stable upon storage. It may be odorless or with odor. Cheap. Immediately penetrate and kills the pathogenic microorganism. Some mild antiseptics are non-toxic and can be applied to soft tissue and skin. Ethyl alcohol, iodine, soap. These are disinfectants. In day-to-day life, we are using these disinfectants. Strong disinfectants are toxic to man but suitable to plant and animal to avoid growth of microorganisms. So called as a sterilizing agent and used as a sterilizing agents. Then some chemicals or disinfectants are used for sterilization. We will see one by one. Phenols. It is widely used disinfectant and antiseptic. Today phenols like cresols, xylenols are used as a disinfectant in laboratories and hospital. They are poisonous for bacteria and virus. Then alcohol are most widely used disinfectant and antiseptic. 80% alcohol is used as a sterilizing agent and widely used to inhibit growth of virus, bacteria and fungi. In laboratory during tissue culture practice, the skin of hands sterilized with the help of alcohol. Next chemical is halogens. Halogen in different 5 elements is used as a sterilizing agent. It is any of the 5 elements: fluorine, chlorine, bromine, iodine, astatine. The halogens iodine and chlorine are important antimicrobial agents used as a disinfectants. Iodine is also inactivated by organic matter and powerful rapid skin disinfectant. It is used for preparation of skin for surgery. Iodine have a wide range of antibacterial, fungicidal, antiviral activity with rapid action. It is able to reduce disinfections caused by pathogenic spores. Then chlorine is used as a disinfectant for municipal water supplies and swimming pools and also employed in the dairy and food industries. It may be applied as chlorine gas, sodium hypochlorite, calcium hypochlorite. It is widely used as sterilizing agent for the control of bacteria and fungus. Chlorine solutions are used to make very effective laboratory and household disinfectants. Then formaldehyde can kill all organisms. For example, spores. It is cheap and it will not damage fabrics or clothes. It is used in gaseous form. It is dissolved in water or alcohol before its use. It is widely used as disinfectant in hospital and laboratory equipments. Next chemical, mercury chloride, HgCl2. It is poisonous chemical substance used as sterilization of glassware experiments or glasswares equipments which are used during different experiments are sterilized with help of HgCl2. It is used as surface sterilization of living tissue, but in very low concentration it is useful. Glasswares are dipped in distilled water containing mercury chloride during the sterilization process in the laboratories. Then H2O2 or hydrogen peroxide is widely used as sterilizing agent. It is used in surgical instruments in the hospital and skin disinfectants. Thank you.